let's have a short video lecture on basic steps of cloning and its enzymes. What is cloning? Cloning is nothing but production of similar type of clones which are genetically identical. And what are the enzymes? Enzymes used in cloning are called cloning enzymes. We'll know them in a further slide. And so let's start our video now. How does a protein synthesis takes place? Let's start with a cell. Cells are present in a greater amount, whereas now we take a single cell which consists of nucleus. The cell which consists of a nucleus is called a eukaryotic cell, where the genetic material is present in nucleus. Whereas if we take a prokaryotic cell, the genetic material is just dispersed in a cell without proper arrangement. Now we'll take a eukaryotic cell which consists of a nucleus and the genetic material is present inside it. So now how does a protein synthesis takes place from DNA? DNA transcribes to RNA and then RNA translates to protein, and this is how protein synthesis takes place. These two steps of transcription and translation are very important, plays a vital role. So now there are many enzymes, factors and enzymes which helpful in both this transcription and this translation process, and all the central dogma depends upon this transcription and translation processes. So now let's start with steps involved in cloning. Later we'll discuss about what are the enzymes involved in cloning. So there are 5 steps which are present in cloning. That is, firstly, a gene of interest to be selected, and later a vector need to be selected where our gene of interest will be incorporated, and later this makes a recombinant molecule. Now this recombinant will be transferred into our host cell with the different types of transfer methods, and then after introducing our recombinant into a host cell, then the screening methods comes. Screening methods is nothing but to know whether our recombinant is surviving in the host cell or not. And there are different screening methods also which will be discussed. I think this slide might give you much more clarity about the cloning process. Now, the first step is nothing but selection of gene of interest. Selection of gene of interest is nothing but there is a long stretch of DNA which is present, from which we have to take a short stretch of DNA which is interested of study, and this is called area of location where we have to study. With the help of restriction enzymes, we have to take our gene of interest. This comes with the first step, and next comes the selection of vector. Selection of vector is nothing but we need a suitable carrier molecule to carry off gene of interest, and they both combine and then go into the host cell. Okay. Now plasmid with the help of restriction enzymes, here a vector will be made a cut where our gene of interest goes through here. Now combination of both our gene of interest and vector is called a recombinant molecule. This is step number 3. This recombinant molecule is formed by the combination of both our gene of interest and vector, and with the help of ligase enzymes, the ends of it will be closed. So now we got a recombinant molecule which is a recombinant DNA. And there are other several names for this. Hybrid also can be called. Okay, now this gene of interest is introduced into the host cell. Host cell is the required media where our recombinant will be divided and produces more number of clones. Now after the insertion of our recombinant into the host cell, later produces our clones. Okay, I'll just tell you what are the screening methods. Actually, this is our host cell. When our recombinant is given into the introduced into the host cell, then there might be 2 chances. The chance one of survival. Survival means If a host cell treats a recombinant as a self and it lets it to grow, so this is how our process is successful where clones are produced. If the recombinant is treated as a foreign particle, then our process is not successful where our clones are not produced. So this is how the complete process of cloning. These are the steps involved in cloning. And I think you might have got there are different enzymes present for cloning. In this we have discussed about ligases and restriction enzymes. So we'll have a brief idea about it in the later slides. In step number 2, we have discussed that there need to be a vector or a carrier molecule to hold our gene of interest. So, what are the minimum requirements for a vector? First is origin of replication, restriction sites, and one marker site. It should consist of at least one restriction site and one marker site and one origin of replication. What are the uses of it? Restriction enzyme digest at the site of restriction site. So that's why we need a restriction site. Later coming is we also need a marker site. Marker site is used for screening methods. According to this, a different vectors can be selected. For example, plasmid, cosmid, etc. According to our requirement, we have to select the correct vector. And here you can also see our gene of interest which holds a vector. So next let's move into the enzyme. These are the different enzymes which are nucleases, polymerases, ligases, DNases and RNases, transferases, kinases and phosphases, and restriction enzymes. So let's start about nucleases. Nucleases are the type of enzymes which shows their action on phosphodiester bonds and never on hydrogen bonds. Okay, this is done about nuclease. And later moves, we all might have it out, why do we select a nuclease instead of a restriction enzyme? I have an answer for you. This is, suppose if our restriction enzyme is not available with respect to our gene of interest, or it might be cutting at our gene of interest and disturbing our gene of interest, then in this situation or in a different type of situation we go to nucleases. And I have told you these break down phosphodiester bond and never on hydrogen bonds. For example, if we take a double stranded DNA, never a double stranded DNA is made into a single stranded DNA, because this is the position where hydrogen bonds are present between two strands. So I have told you that nucleases show its action on phosphodiester bonds and never on hydrogen bonds. When these hydrogen bonds are not disturbed, so there is no breakage of hydrogen bonds, so the actual double stranded DNA be like this and never it becomes like this. Okay, and how are the phosphodiester bonds formed is through a different enzyme. We'll discuss it later. And in nucleases, we have two types of nucleases, exonucleases and endonucleases. And also there is another type, further these exonucleases are further classified into three types: BAL31, EXO3 and lambda EXO. The application of these three are different. For example, our gene of interest is located in the middle here, then BAL31 is used. If our gene of interest is located at end, this is 5' to 3' direction, and if our gene of interest is located here, then we used lambda EXO because it shows its action in 5' to 3' direction. So by this what we have understood, if our gene of interest is present to the 3' direction, then we have to use lambda EXO, because It shows its action from 5 prime to 3 prime direction. Like it starts chopping like this. And it should stop at our gene of interest because it should not disturb our gene of interest. Similarly, this XO3 is used in 3 prime to 5 prime direction, and when our gene of interest is present in 5 prime direction. When our gene of interest is present in 5 prime direction, as its action is from 3 prime to 5 prime direction, it starts chopping from the 3 prime direction and never disturbs our gene of interest. So I think you have got an idea about exonucleases. Next, coming to endonucleases. Before going to endonucleases, I have told you that BAL31 is used when our gene of interest is located in middle of a stretch of DNA. So you all might have a doubt. So why it is a type of exonuclease? Because exonuclease's action is on ends only, and endonuclease action is in the middle of the genetic material. When our gene of interest is located in middle. So exonuclease's function is, as we know, it is at the ends. BAL31 has action of combination of both XO3 and lambda XO. So a cut is made like this, here with one enzyme and here with one enzyme. So need not require both of these enzymes. So this is why we use BAL31. Moving to endonucleases. Endonucleases is used when our gene of interest is present in the middle. And the examples of restriction enzyme, DNase and S1 nucleases. And S1 nucleases are single stranded DNAs, and double stranded DNases and restriction enzymes are used. DNase does not have a specific function. It just chops like normally how it cuts away, it goes on like that. So let's move to our next slide, which is polymerases. Polymerases is mostly used in the process of replication because it forms phosphodiester bonds. Now phosphodiester bond is produced if there is a phosphate group at 5 prime direction in 5 prime end, and in 3 prime end if there is a OH group, then a phosphodiester bond is formed. Okay, this is done during replication. There are different types of polymerases like DNA polymerase, RNA polymerase, reverse transcription polymerase, tag polymerase, and etc. So now the mostly used are DNA polymerases. There are 3 different types of DNA polymerases: DNA pol1, DNA pol2, and DNA pol3. Their action is different based on nucleotides. If we take DNA polymerase 1, it adds 20 nucleotides per second. If we take polymerase 2, 30, whereas polymerase 3, it adds 50 nucleotides per second. So now, DNA polymerase 1 and DNA polymerase 3 are most preferable because it has action in both 3 prime to 5 prime direction and 5 prime to 3 prime direction. These both DNA polymerase 1 and DNA polymerase 3. Whereas if we go to DNA polymerase 2, its action is only in one direction. So mostly they take DNA polymerase 1 or DNA polymerase 3, and also it adds more nucleotides per second, DNA polymerase 3, which is a good sign. So next, RNA polymerases are also used. Next, there is a reverse transcription polymerases. Reverse transcription polymerases is nothing but, we all know that mRNA to protein with the help of DNA. This is transcription. This is translation. In this process is a normal process, whereas Here, mRNA is converted back to DNA. This is called cDNA, which is called a reverse transcription process. And the polymerases helpful here are called reverse transcription polymerases. Next comes is the Taq polymerases. Taq polymerases are the type of polymerases which are used in higher temperatures. Normally, there might be enzyme which work till 65 degrees Celsius, whereas Taq polymerases are nearly done its action at 92 to 95 degree Celsius. Depending upon collection of species from different areas, they are commercializing the product according to their commercial ways. Coming to this polymerase, it has an action of dual function, which is combination of reverse transcription polymerase plus DNA polymerase. This is the polymerase used. T7 polymerase is used at viral DNA or special type of DNA molecules. Next comes transferases, which used to be a part of polymerases, but whereas due to its action it is separated from this polymerases and it has given a different group now. Let's move further. Ligases. We all know that where ligases are used. Ligases is used during recombinant formation to join both the ends. Now, there are two different types of ligases, DNA ligase and T4 ligase. It depends upon cohesive ends and blunt ends. If cohesive ends are, both the cohesive ends are there, then DNA ligase is used. When both the blunt ends are there, we use T4 ligase. What is cohesive ends and what is blunt ends? If there is an action of some enzyme and if the cut is made like this, then it is called cohesive. Whereas if the cut is made like this with equal, then this is called blunt and this is called cohesive. This is cohesive ends, then this is blunt ends. And next is DNases and RNases. DNases and RNases are not specific and they just chop or digest randomly. Okay, moving. And next comes the restriction enzyme. Next is restriction enzyme. Restriction enzymes are used to digest at a specific site. We all know that. Restriction enzyme should have a target site which is specific. For example, if you take EcoR1, it shows its action only on GAA-TTC and only its target site is here between G and A. So by this it has a cut here in 5' to 3' direction and 3' to 5' direction like this. So by this we get a cohesive end. Like this. Whereas there are some types of enzymes which have different target sites. Like if we take HIN2, it produces a blunt end, which is like this. This is how a cohesive end or blunt end will be produced with the help of restriction enzyme. Okay, these are called molecular scissors because it is used here in genetic engineering processes mostly. And coming to the next is it requires ATP and metal ions. Metal ions can be calcium, sodium, etc. So now the thing which uses a low number of metal ions and low ATP is the best to be selected. So restriction enzymes plays a very vital role in this processes. And thank you. I think you have got a brief idea about what is cloning and what are the enzymes used.